We will get acquainted. We will get acquainted with the different domains of bioinformatics and learn more about the omics studies and technologies. My name is Sonalika Ray, and I'm the OmicsLogic Community Manager responsible for daily interaction with thousands of users that are a part of a bioinformatics community managed by Pine Biotech. OmicsLogic is an international program that is running in 5 different regions with over 10,000 users around the world. Due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and leverage our online training resources, adapting them to the needs of the students and researchers around the world. Our training is designed to allow participants to apply methods to curated and new data sets and develop independent projects that showcase what has been learned in the context of a personal portfolio of projects. And as a result, anyone starting on this journey in bioinformatics can start developing a project of their own. In fact, we have already worked with many students who have completed this kind of training and developed interesting research projects that won awards and got published in peer-reviewed journals. And today in this workshop, you will be given the directions to access the T-Bioinfo platform for bioinformatics, processing, and analysis of transcriptomics data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipelines. Different stages of analysis are performed in different sections on this multi-omics platform. The T-Bioinfo platform is built to help prepare biologists and clinicians for the large multi-omics data sets they have to deal with on their own without relying on technical support to set up computational environments and pipelines for processing. The simple to use solution can help train and prepare anyone with a biological data set for advanced computational analysis, visualization, and annotation. To learn more or try the T-Bioinfo platform, you can register yourself for a free demo account and connect with one of our representatives. I also want to share with you that we have an upcoming Omics Research Symposium that we would be talking about in more detail. The Virtual Research Symposium is an opportunity for students, faculty, and companies to come together and discuss advances in bioinformatics. In this session, speakers will highlight advanced new areas for bioinformatics as a discipline, offer students and faculty insights into best practices for mastering bioinformatics skills, and allow them to explore career opportunities in academia and industry. We will cover bioinformatics as cutting-edge research and applications for undergraduate, master's, and PhD programs in bioinformatics. We will also have recruiters, industry internships, and talk more about job opportunities, effective resources for training and education. And we have amazing prizes for best school, college, and citizen research projects as well. Now, to discuss more about the workshop and also about the symposium, I would like to pass it on to CEO Pine Biotech, Ilya Brodsky. Over to you, Ilya.
Thank you, Sonalika, and welcome everyone to today's webinar. I'm glad that you were able to join. And what I would like to start with today is to briefly discuss what are some of the challenges and opportunities in this domain, because as we all hear, there are huge opportunities with novel technologies, with precise data in all of these different areas. I mean, we all heard about the revolutions with mRNA vaccines, CRISPR, genomic surveillance, single cell data. All of these also come with computational technologies like the growth in artificial intelligence and machine learning, quantum computing, high performance computing, cloud technologies, as well as big data and integration. So many of the people that come from the life science background, from biology, from clinical background, from biochemistry, when we hear about these advances in computational technologies, there is a gap that prevents us from directly using a lot of those techniques because it requires engineering skills or data analysis skills. And that gap creates a lag behind the growth in the amount of data and the quality of data. and the information, the actionable information and the insights that this information can provide us with to actually do something in the fields of biology, medicine, biotechnology, and agriculture. In fact, in a survey in 2017, there was a question about which step of one of those techniques, RNA-seq, was the most intimidating, and the answer was that the majority of the people struggled with the analysis of this data, right? So as you can see, 49% of the responders said that analysis of RNA-seq data was the most intimidating step. And one of the things that we realized as we started dealing with a lot of people interested in bioinformatics is that there's a whole range of prerequisites that students oftentimes face before they can even get started. And there's also a lot of hands-on experience that you need to acquire as you're trying to analyze different types of data, which makes it also difficult to analyze larger data sets, because on your laptop, on your computer, oftentimes without great access to internet, large storage, a lot of processing and RAM power, you just cannot analyze a lot of these newer types of data that are coming out. So that's why what our team did is we decided to build a community to address this challenge. First of all, this community is to provide training in bioinformatics. It is also to bring together experts and those who are passionate about data-driven research and data analysis. And the objective is to really enable students, clinicians, and faculty without the engineering background, without the technical skills, to be able to leverage these large data sets and apply analytical tools to complex data. Now, we realize that this gap doesn't appear by itself. It's a problem that a lot of universities, colleges, and other research institutions are trying to address. And the gap that is created at the university, at these places of learning creates confusion, because on one side, you get excellent training in technical skills, and you probably have some expertise at the end of receiving your degree. But at the same time, there's a confusion about how does this actually work? How do I actually manage to use these tools effectively myself without having that practical experience? And so to address that gap, we started putting together the pieces: the big data analysis infrastructure, a network of experts that can assist and help and mentor and guide students that are interested in this, as well as our training and research support infrastructure. So to show you how it all works, I would like to showcase one of the central pieces where we try to connect all of these things together, the Omixlogic Portal. So if you could please go with me to the Omixlogic Portal. If Sonanika, you could please post the links in the chat. So on this portal, first of all, let's take a look at the home page. You will see here all of the different pieces organized together. So it's a free registration. Everybody can register today. The way you can register is click right here on this button, register for free. And you will be taken to a page where you can register yourself using your email or one of the social media accounts. Now, if you don't have an account, you click right here, create an account. I'm going to give it just a few minutes for everybody to go through the registration. After you have registered yourself, please put number 1 in the chat so that we know that you are registered. And I'll go back to the home page and just wait for all of you to let me know that you are registered. All right. Thank you very much. All right, great. Okay, so I see quite a number of you have already registered. So thank you for putting that there. So let's take a look at what is the OmicsLogic Portal. The OmicsLogic Portal seeks to bring together everything that you need to get started and grow. This is relevant for students as well as for faculty, and today I will demonstrate how. First piece is that we realize that those coming from a life science background are interested in bioinformatics primarily because it is a way to address interesting projects that are at the cutting edge of oncology research, infectious diseases, neuroscience, and agriculture. These are the precision medicine, precision agriculture, novel interventions, novel therapeutics, novel diagnostic methods. So these methods rely heavily on data. The data is complex, and the data can reveal important insights to drive each one of these fields forward. So that's why, according to these different types of projects, we created a case study driven approach to understand the value of omics data and practical steps to analyze this data. First of all, understanding the logic of the analysis, and then also acquiring practical technical skills that if you decide to go that route, you can master right here on the portal. A critical piece of this experience is the T-Bioinfo platform. The T-Bioinfo platform is a place where you can analyze, integrate, visualize multi-omics data. Okay, so once you have created your account, in 24 hours after your account has been created, you will be granted a demo account on the T-Bioinfo server. Okay, so to demonstrate what the server is going to be useful for, you can click right here. Now, if you already had an account for a while with us on OmicsLogic, you will be able to log in here as well. If you don't have the access just yet, you will have to wait for 24 hours before that account is generated. And in any case, after you've created a free account on the OmicsLogic portal, you will have a demo account here, which is very useful for education. Now I'm going to demonstrate to you today how. So, and thank you, Ojasvi, for posting the link. So on this link, you will find an areas of analysis section. Now, there's a lot of different types of omics data, and there's a lot of things that you can do with this data. But let's start with some of the basics. Let's understand just two examples of what you can do here without any coding, and how does this fit in to the OmicsLogic portal experience. So we'll take a look today at, first of all, one of these sections. So you can see for next generation sequencing data, one of the data types is transcriptomic data analysis. And here, right under this RNA-seq chip, you can open up some demo example projects that you can run right here on the portal without having any engineering or coding skills. So an RNA-seq pipeline typically contains several different logical steps. If you click right here on start, you'll see those steps highlighted in different colors. Pre-processing, which means cleaning up the raw data that is produced by the sequencer. Mapping, or trying to understand which piece of the genome did the read come from. And then quantification. How much reads do I have in each segment or a gene or an exon, so that I can understand what are the levels of gene expression. To understand how these pieces connect together, we can actually build a pipeline, which is just a series of algorithms connected together to understand what are the inputs and what are the outputs. So after you finish reading through this first pop-up, you can also open up the input files and the output files. Okay, and so here you can click on OK. And now a limited selection of options is highlighted based on your previous selection. So since we have a project that we loaded in, it's a project on the transcriptomic analysis of cell lines from different types of breast cancer subtypes, we now have to pre-process or clean up the reads that came from the sequencer. To do that, we can use Trim-O-Matic, which trims adapters from each side of the reads, and PCR Clean that reduces the artificial amplification based on PCR bias at the step of bridge PCR amplification. So to address this challenge, we have a special algorithm called PCR Clean. After we've cleaned up our raw data, remember, we need to map it onto the reference genome. Now, there's a lot of different options here. You can see that they're organized into logical groups. And within each group, there are several things that you can compare. So essentially, you can benchmark and compare how the different algorithms perform and understand the inputs and outputs for each one. So let's use the simple option. We'll use Bowtie2, which is an algorithm that provides us with capabilities to rapidly map all of these reads to the reference genome, understand where they came from, and create a mapped file or a BAM file. Okay, so now we're going to connect this to our previous steps. And finally, we have to quantify the read. So what is quantification? Ultimately, we want some numbers. These numbers come from an abundance of reads, which are fairly short, on a long sequence, maybe thousands of nucleotides at a time. And to understand what is that number that we need to assign to the level of expression, we need to use a complex algorithm. In this case, we're using RNA-seq expectation maximization, or RSEM, which actually assigns numbers in a few different formats: FPKM, RPKM, TPM, or raw read count. So in this case, we're going to connect RSEM to the end to complete our pipeline. And once this is completed, you will quickly see here some outputs that are useful to understand what transcriptomic data is all about. So the transcriptomic data, as you can see right here, provides us with an expression of exons, an expression of genes, and an expression of isoforms. Expression of genes are the approximately 22,500 genes that we have in the human genome. Each gene, as you remember, is combined from different exons, different combinations of exons. So we can look at all of our exons, or we can see how those exons are combined into actual transcripts or isoforms of genes. And so we can download these tables and just see what these tables look like. And so that is the first important application of this T-Bioinfo platform. Right here, without any coding, we were able to take some raw data, prepare it for analysis, analyze it, quantify it, and produce a table of expression. Now, the second question, right, as I already said in the beginning, as people who come from a life science background, many of us are interested in different types of projects where this technique could be applied to. So that's why on the Learn portal, if you go right here to the Courses tab, you will find several projects that are used as case studies to give us access to curated data sets with an explanation of how step by step we can go through those transcriptomic analysis steps and get some insights, get some meaningful understanding about the data. So let's take a look at one of these. I will open up project number 5, Modeling Cancer Precision Medicine. This is a project where the data came from. So we looked at a data set. This data set is explained right here in this project. I pasted the link inside the chat. So the first thing that we can do is we can go to this first lesson. And this lesson is, by the way, free for anyone that would like to use the portal to try it out. You can see all of this information that I'm showing you. So the project explains how patient-derived cell lines that came from patients with different subtypes of breast cancer were used, these cell lines were used to generate multi-omics data so that we could try and link signatures in omics data with another type of data, which is measurement of drug efficacy. The measurement of drug efficacy was done with a special way to quantify how a cell line responds, how a cell culture responds to treatment, and that is called LD50. So now we have signatures from the transcriptomic data that we have to find, and based on those signatures, we can predict which types of treatments are going to be the best for that patient. So as we go to our next step, we can read about what you can learn from this case study. There are several different things that you can learn. First of all, you can learn about different types of data, genomic, transcriptomic, and even the microbiome, metagenomic. So you can analyze different types of omics data. Number two, you can see the application of machine learning in this example, because to understand what signatures are specific to each subtype of those breast cancer patients, we can use a classification model, which I will show you in just a few moments, of how you can train such a model right there on the Bioinfo platform. But also you get exposed to a number of concepts in precision medicine, which go to explain biomarker discovery, integration of multi-omics data, essentially to make a decision, a clinical decision on how to treat a patient with the best outcome for that patient. Okay, it's a model, it's not a real clinical project, but nevertheless, it's a model that we can learn from. Now, to learn about the full publication, there's a link right here in number 3 for associated publication that describes how the data was generated, what were the objectives, and ultimately what did the authors use to train their model, validate the results, and propose an algorithm that essentially could be used in a clinical decision-making scenario. So once you complete this step, you can click on next and learn a little bit about how these types of projects are applied in modern precision medicine kind of research, in biomedical research, and what are some of the applications of such projects as we are using today. So now we can complete the lesson. We've understood what is being done here. Okay, I think just need to complete my name here. Okay, and this kind of feedback, by the way, that you submit in the form is very useful for us as we try to improve a lot of the materials on the site. So after we've understood the data and where it came from, next we need to analyze it and prepare it for machine learning. So what is machine learning? Let's go back to this section for the Bioinfo platform. Here we have processing pipelines, right? These are pipelines designed to take raw data and turn them into a structured data. The structured data typically is a data frame or a table. Once we have that table that contains many rows, in our case, 22,500 rows, because we have all of the genes from the full transcriptome based on these cell lines, and multiple samples. In this case, we have 52 samples coming from the cell line experiment, organized into multiple groups. Those groups could be referred to as classes. One of the things that we can then do right here on data integration and modeling side is use data mining approaches to extract meaningful insights from this type of data that we have. Now, there are two major areas that we can apply here. There's unsupervised analysis, and then there's supervised analysis. Before we jump into that, let us download the table that we generated in the RNA-seq data. So I will go back here, and I will again repeat the steps for this demo RNA-seq cell line project. And that will help us understand again how these steps fit into the final output that we have. So we go from start. Again, remember we have 3 different steps: processing, mapping, and quantification of expression levels. Now, if you want to understand how to connect them together, this is an example pipeline that you can see right here. First, we go to pre-processing, which is the blue So we go from trim-o-matic, trimming the reads from sequencing adapters, to PCR clean, where we remove this artificial and biased PCR amplification. Then we have two options for mapping. We can either do complex mapping, where we are detecting exons and preparing our own transcriptome, or we can do simple mapping, where we just follow the instructions in the existing GTF file. We'll take the simpler option today. And then we have to quantify the levels of expression. As I explained already, a lot of challenges with trying to figure out how a certain profile of abundance could be converted to a single number that represents gene expression. And finally, after we end, we can see the output files, and we have here several outputs. From all of these outputs, let's take the cell lines expression data normalized and annotated. We'll download this and we will see what is in the file. Okay, so that we can see what this could be used for when we talk about machine learning. Now, in addition to this, we can download this file, cell lines clinical subtypes, and that will give us a sense of what clinical subtypes are represented with these cell lines. Okay, so here you can see that the names of the columns are names of cell lines. The names of the rows are genes. NSG, right? Those are genes. And we have about 7,000 because this data was already cleaned up from genes with 0 expression and with low expression. In fact, it was cleaned up for expression under 6, and then all of the data was normalized. So all of the data was prepared for downstream analysis. In here, we can see that for each name, each cell type name, we have right here a transcriptional subtype, which means a clinical class that we can use to determine differences between different subtypes of breast cancer. Okay, and so here you see you also have a basic subtype, and then what is the cell type? And all of these cell types are breast cancer cell line. Okay, so what can we do with this information? First of all, if we just take all of these and we put them here, okay, we're just going to see how many do we have, right? We're going to transpose, and let's go to the bottom. We can see that up until here, it looks like what we have exactly matches what we have in the annotation, okay? So we can just take the transcriptional subtype and we can create here another row. We'll call this row group. And we will put in here this annotation. Now, for each one of our cell line names, we have a transcriptional profile. Let's save this. Okay, I'm going to save this cell line expression data normalized and annotated. And the first thing that I would like to do after I've done this is to understand what is going on in my data. What are some of the major trends in my data? The way I can do this is I can go to unsupervised analysis, upload my data, press on continue, and use principal component analysis. Now, what is principal component analysis? I'm sure that many of you already know principal component analysis and what it does, but for those of you that are maybe new to this, I will just briefly explain. Principal component analysis is an analysis of variance of all of the data to maximize variance between different samples in my data and show us the major trends based on all of the features without any feature selection using dimensionality reduction. What does that actually mean? When we look at our data, we can visualize the variance between each one of our samples using 2 dimensions. So let's take these 2 dimensions, right? I can just insert here a scatter plot using 2 dimensions, and I will get some cloud of dots. This cloud of dots represents all of the samples that I have based on 2 dimensions, X and Y. So here I have the X and here I have the Y. And that shows me some trends in my data. But I have close to 7,000 dimensions. So how can I translate this data into a simple visualization that will show me what are the major trends? To do that, there's a mathematical technique that can deal with so many dimensions in a way that we can't visually. And the technique goes like this. If I have a complex shape, that shape has variance. It could be more stretched out or more condensed. If I want to find those areas where it's stretched out, the directions where it's stretched out, I can make a projection and use that projection to explain variance. Okay, so I can make a projection, use it as my axis, make another projection in the second most variable direction, and use that as my second observation, right? And so I can transform the 7,000 dimensions that I have here into just 2 dimensions. And that's why this method is called dimensionality reduction. Dimensionality reduction simply means that I simplify the representation of the data to understand variability overall. So now we have the results of this pipeline. We ran PCA and it produced for us a plot, a visual result that we can understand. Okay, so let's take a look at this visual result. What we can see is that based on all of the data that we loaded into the pipeline, there are 2 major groups that separate the cell line information of all the gene expression. And we can see down here the annotation by group. For example, on this side, we have luminal, and on this side, we have the rest. We have right here, basal, cloud and low, and normal like. So all of these are very different from these. That is new information that we got from analysis that we just performed. But there's some additional information here. For example, we also see how much variance does this explain. And we can see that together, it is under 40%. And what that shows us is that this particular data set has a lot of additional noise that does not clearly explain all of the variability between these subtypes, even though it does have some major trends. An additional analysis that we can perform under unsupervised analysis section is called clustering. So let's try to do that together. Again, we will upload the data set. This time, I am going to remove this annotation. I don't need the annotation for groups. I'm going to remove it. Actually, what I will do is I will put it right here. I don't need necessarily to know the names of my samples. I want to understand what groups can clustering put all of this information in based on my clinical annotation. Okay, and I will just save it again. Let's upload this data right here. Continue. Okay, going to be processed. All right. And now we will perform clustering. So you can see that the first section is visualization. We just visually want to understand what's going on. The second section is called clustering. So clustering helps us identify specific groups of data, but also define the relationship between them so that we can understand in greater granularity what groups are found in this data overall. So the way to do this is to use something like hierarchical clustering with linkage. Right here we have linkage called Ward D2, so we will change it to that. We'll use Euclidean distance. Again, if you kind of imagine this cloud of dots, we can just measure distance between each dot and understand how closely they are related to each other. And right here, we need to put in how many clusters are we actually expecting. And since we know that the total number of the subtypes is 4 and not 5, let's put in number 4. Now, do we need to transpose or not? Transposition of the table means that we flip it on its side. Right now, our column names are samples and our row names are genes. If we analyze the data as is, it will produce clustering for the genes that we have. But instead, we want to cluster the samples. And so that's why we need to transpose the data. So this should be changed to true. Click on save and we'll name it. As it's performing clustering, let's just briefly take a look at what are some of the other options that we have here. So first of all, we talked about PCA. PCA is a very useful tool for us to use, but a lot of times PCA will perform differently on data that is normalized or non-normalized. Remember that we took a data set that was already curated. Somebody messed with it, cleaned it up, normalized it, so it's easy for us to use in this example to learn about the methods and what they do. But if you actually want to pre-process them, we have an additional section right here under areas of analysis, under utilities, that can help us prepare the data specifically for the type of analysis that we would like to do. We have quantile normalization, batch correction, missing data approximation, and other types of methods to prepare the data for what we just did. Beyond that, we have different other methods of visualization, including box plots, heat maps, t-SNE, APE, and others, right? So there's a lot of other options here. So typically when you go through the project represents a curated data set that already prepares the data for simple use. But as you go through these different examples and you understand the logic, you will also learn about the detailed steps to run data preparation, data wrangling, and other tools. And I will get to that in just a few minutes. Let's see if our clustering is done. It is. So let's take a look at the output here. Again, we have a number of different useful outputs. First of all, we have only the prediction of clusters. That means we can say which sample was assigned to which cluster. The second thing is we have the full table. Not only do we have the clusters, but we also can visually see all of the gene expression values as well. And finally, over here, let's take a look at this PDF. It's a visual output that will clearly show us what is the data looking like in terms of groups. Okay, so let's take a look at what's happening here. As we saw in the previous PCA, we have two major groups, a luminal, and right here we have all the rest. But if we look inside all of the rest, we can see that cloud and low right here is a separate cluster. Basal is a separate cluster. Normal-like is a separate cluster. And then here we have some outliers. Here we have some samples that look differently from the rest of their group. But here's another interesting insight. Right here we have luminal as a separate group, and right here we have another separate group of luminal. What do you think are these two luminal groups that we found in this clustering? I'll give you a moment to paste your answers in the chat. Okay, what do you think is the reason that we see two major groups within luminal? Okay, based on distance, yes. But why do you think we have two major groups? So for those of you coming from a cancer and oncology background, you already know that there's luminal A and luminal B, right? Even though we don't have that in our clinical annotation, it is clear that also here we see two major groups of luminal. So when oftentimes the clinical information that we have is based on some specific markers and not all of the data. But if we take all of the data into consideration, we will see that actually a more true biological representation can come out from mining the data, okay? So another important thing that we see is that this luminal sample is an outlier. And that means that we can probably address this before we go into the downstream interpretation. Okay, so Kira Tang is saying each of the two major groups contain those that are more closely related to each other. Exactly, they actually form subgroups within this major group of the luminal subtype. Okay, so there are some interesting things that we can learn right here. Now, as I mentioned, there are many other things that you can do with this information. Now that we've understood the data, we can do multiple things. One of the things that we can do is differential gene expression. Differential gene expression can provide us with information about differences between two groups, okay? Outliers are not necessarily wrong data, but they could be either, if you can imagine a group of people, there's always somebody strange, right, in that group of people. So that strange person is an outlier. Now, what's wrong with that person? We can say, like, is that a wrong person? It's not a wrong person, it's a unique person, right? They're special, they're different. Maybe if we want to understand the major trends in the population that we're studying, they're not a good representative. But if we want to understand what is wrong with that person or how are they different, we can ask that question, right? So this type of exploratory analysis is really designed to give us insights, give us new ideas that are data-driven. By looking into the data, we get new ideas, and now we can ask questions. How do we ask questions? Well, one way that we can ask questions is we can say, let's forget about everything else, and let's compare. Here we have luminal A, here we have luminal B. How are they different, right? If we want to analyze differences between two groups, we can use differential gene expression. So differential gene expression consists of several different steps. Number one, we have to understand statistically significant genes that are differentially expressed. That is done by different algorithms like EdgeR and DESeq2, which provide us with essentially two important values: the significance, or a p-value or an adjusted p-value, and how much are they different between each other, which is fold change. So once we have that information, we can then perform two additional steps: enrichment analysis, or what types of pathways and gene ontology terms are enriched with genes that are significantly differentially expressed, or gene set enrichment analysis, which is a similar approach that produces an annotation of visual pathways, different types of metabolic pathways, signaling pathways, to help us understand how do those genes actually participate in the processes that we can interpret biologically. So the other type of a question that we can ask is, what are the unique genes that have a profile or a signature associated with a particular group? And that is called classification. Now, classification is very useful when we want to train a model, make sure that it works on our test data, and then use it for big data analysis, right? So for example, I have a population of patients. I've studied them and I've understood how they respond to different treatments. I have a new patient coming in. How do I actually assign that person to a group that I understand? One of the ways I can use gene expression data, classification, and find who he is most similar to. And another thing that we can do here is we can do what's called feature selection. So feature selection will allow us to select specific types of genes or markers that I can use to look for when I have a new clinical challenge. So we're not going to do this here, but I want to show you one thing that I didn't show you up until now. In each one of our outputs, okay, let's take a look at this PCA. One of the outputs that I have is a package of the R scripts that are useful to analyze this data independently. Because one of the objectives that we have on the Learn portal is not just to tell you about what analysis you can do and how to use the curated data sets, but it's actually to go beyond that. And if you would like to acquire the skills for coding, you would be able to do that right here on the portal. So let's go and take a look at one of those courses that are going to be taking this further. Sorry. Okay, right here in the search, you can type in data science, and you will see that there are two data science courses: Introduction to Data Science in Python and Introduction to Data Science in R. Let's go to Introduction to Data Science in R, and I'll briefly show you how this works. Okay, so this is the link. Just again, paste it right here in the chat. Again, every single lesson or every single course here includes some free lessons that you can go through to understand how this portal works. Okay, today we will use one of these free courses called Dimensionality Reduction, PCA, and Visualization. So what is principal component analysis? I've already described. We can take all of our data. As you can see, I have the names of the cell lines. I have the annotation by group. Then I have my data in the cells for every gene included in this little table. This table is a small selection of genes that we're going to quickly play around with right here in the browser. Let's go to next. To understand what PCA is, there's a short video and some code that actually explains what are the steps that you take to go from having that full table to running a principal component analysis. So first of all, there's a library. We can load a library that somebody wrote of a bunch of different functions that we can use to analyze this data. We then can load the data, giving it a name and putting it into an object, transform or transposing our data just like we did in Excel, but right here we're doing it in R, and then running PCA. Now, to run the analysis is very simple. All you need to do is call a function from this library. You don't have to write the function yourself. You can just run it. So let's try and run it right here in the browser. We'll click on next. And here we have a little window that opens up where we can run and modify code. Okay, so we have the input file. As you can see, the input file is for only 15 genes. We then define the data. We transpose the matrix, and then we run PCA. Okay, so let's run this. Okay, all we needed to do is run. We have the output right here. Okay, and we have a plot. And this plot shows us multiple groups, not just two, but it shows us 3 different groups that we don't know what they are, right? Because there's no annotation. And here we have another version, again, no annotation. So one of the things that we would like to do is to actually run it in a way that we can understand what's going on. Okay, so now we can't do that because this is an assignment. So we learn how to run it right here. Right here, we need to start thinking, how do we change this to make it work? A typical way to think about that is to read right here and see what the problem is. Okay, so what it is saying is that just by itself, this is not going to work. Okay, so what is not working? We need to figure out based on what we have right here. Okay, so try running this code by adding this part of the script to what is already in the code block. So here we have something, and here we need to actually add this right here. Okay, so we need to take this, and you can see right here, there's some open parentheses that are not closed, so we can't do that, right? So we can see samples, call name expressions right here. We just need to take this, okay, and we need to replace all of this. Okay, and we do not need. So when we run this, okay, we have right table. So we have right here, the right table part, right, that we did, say the PCA. So right here, we will write table. And we have plots, okay, so we can just do this. Let's run it. Oh, I did this twice. Okay, define the data. Okay, we already did this. Okay, error running code. Let's figure out what the problem is. Okay, file PCA table. Okay, full table expressions. Line 19. Print length. Okay, so right here, yes, we need to write in what are we length of? Okay, so this is going to be PCA res. Okay, expressions T not found because we have this and we need to actually transpose it, right? So right here, we have the line on how to transpose it. So right here. All right. And the answer is correct. So with joint efforts, we've written everything that we need to run it, but we also want to plot this. All right, so now we also made a plot, but now we see what this plot is based on. And the plot is essentially based on a new table that was prepared for PC1 and PC2, right? And so now that we see what this result looks like, we can actually understand the deeper logic behind the analysis that we are doing. We're essentially translating this table that we started with in the beginning of the 15 genes to another table that has principal component 1 and 2, giving us positions on the scatter plot. Now, we can also perform other types of exploration of this new object that we created, the PCA object, to understand what is included inside. And so here we have the full 3D representation of this principal component, which shows us the principal component analysis results, not only in 2D, but actually in 3D. And here we can see a pretty good separation between all of the different types of cancer that we have. Luminal. However, you can see that one of those luminal guys is right here, right next to basal, which is probably that outlier that we saw before, right? And so this shows us what is the outlier. The outlier is the one that was not like the rest. It was a little bit out from the rest, outlier, close to another types of cell lines, okay? So after this, we can see some additional code and we can go through step by step to really understand more and more how do we take this basic script that we learn how it works and finally produce a plot that we can understand with annotation for groups, even with defined groups that now are according to the classes that we have. And that is all really the objective of the OmicsLogic portal. The OmicsLogic portal connects the logic of analysis of complex omics data with case studies where you can understand the relevance, the need, and the outputs of different types of analyses. You can choose to go deeper and understand the analysis details and syntax of coding, or you can also join a program that will be mentor guided, and someone like what we did today will just go through some of the examples that we are having, and those examples will be used okay, let's go back to programs. And those examples will be used to explain some of these concepts in a more guided way. So all of this information is linked together under my personal profile. And this is an important part of this portal, is that all of the activity that I just did, as you can see, I've completed modeling precision medicine publication. I've completed the step biological concept of precision medicine learn, right? So every step that I take accumulates into a number of points that overall take me from a beginner to an intermediate level to an expert. Once I cross the 10,000 point threshold, I will be able to design an independent project. An independent project is essentially a publication that I can design to showcase what I have learned and take a data set of my own, applying the skills that I've acquired during the training and showing what it is that I have learned. Now, here are some example projects that other people have completed during the training. And once you complete a project, we are excited to show how you can use the experience and the projects that you have created to essentially move forward in your career. And so now briefly, let me introduce you to the Omics Research Symposium that we are planning to start August 21st and 22nd. Okay, we already have over 1,500 participants that have registered for the workshop, or sorry, for the symposium. It will be a 2-day symposium where we will have experts in the field of bioinformatics coming from academic programs around the world, speaking about the need and the challenges and opportunities in bioinformatics and the need for training. And in the second day, providing you with some insights on how to get involved. How do you get involved? You can present your project and get feedback from experts. You can learn about academic programs for undergraduates, high school students, undergraduates, graduates. You can also understand how to participate in our research fellowship program that will help you design and complete a project in a short period of time, between 3 and 6 months. Now, at the same time, you will also learn about different companies that are hiring for bioinformatics skills in Africa, in India, in the US, and around the world. And we'll have some experts from pharmaceutical biotechnology companies and bioinformatics companies that will explain what they're looking for and how to get involved in what they are doing. Yes, students from non-biology background can attend the symposium. The symposium is open for everyone who is interested in bioinformatics as a researcher, as a student, as an expert, as an industry professional. So down here at the bottom, you will find a short video that describes what the symposium is all about. And to summarize and kind of share with you the details of the registration and the schedule, I will pass it on to Dr. Mohit Mazumdar, who will be one of the people organizing some of the panel discussions. Mohit, are you there? Okay, I don't
I
see.
think sir is facing some network issue.
Okay, so Sonanika, maybe you could just take everybody through the schedule to explain what is going to happen during the symposium.
Yes, sure, Ilya. Just a second, let me just pull out the page. So while I'm just pulling out the page, it would be great to know about you all. You can let us know about your research background and what is your research interest. And so this topic today was bioinformatics for non-bioinformaticians. So hoping that you all are from a non-bioinformatics background, we would love to know how come you are inclined towards getting acquainted with bioinformatics and what sort of purpose you had attending the workshop today, and what do you foresee yourself in future. Okay, so talking about the schedule of the symposium, we have great speakers who would be talking about the area of their expertise. And the symposium is on August 21st and 22nd, as Ilya has already told us. I would be sharing the link with you in the chat box, and also I would be mailing all the details with you all on the mail after the workshop is over. Here is the schedule link. So we would be beginning with a talk with Dr. GPS Raghava, and he's the head and professor at Triple IT Delhi. And then we'll have a talk on industry-driven education and healthcare in India by Professor Pallu Radana. And having a short break, then we also have a team in Africa, whom we call as OmicsLogic Africa or Eureka. So we'll be having some opening remarks by them as well, and we have great projects on which our Africa team has already worked on, and we would be having their presentations as well. And then we would be having a talk by Dr. Anita Ghansha on bioinformatics of infectious disease and relevance to public health. And then Ilya would be giving an opening ceremony and topics, telling us all about what we would be going ahead with, followed by a panel discussion where we'll be having Dr. Gus, Dr. Leonid Brodsky, Professor Sona Vasudevan from Georgetown Medical University, Professor Pallu Radana, and Dr. GPS Raghava again. And now what we have done is we have few breakout rooms. So in the breakout rooms, we would be having talks going on in different sorts of fields. So let's say if you are interested in artificial intelligence or machine learning, you could be in one breakout room. Or if you're interested to learn more about astrobiology, that's an emerging field, and we have a lot of projects on that as well, and our research fellows are working on it too. And then we have a field as agri bioinformatics as well. So these are different sorts of field you can be a part of and join the breakout rooms depending on your research interest. You can find the details of all the speakers who would be talking about these different sorts of fields and giving their talk on their expert topics. And then comes the second day, where we would be having talks by Professor Seema, Professor Sona, and Dr. Philip Compu. And we have the details and the topics of their talks already mentioned in the schedule page. I would love if you could go along with me through this schedule page itself and learn more about what all talks you are going to have. So you see, it's really actually because of COVID, this thing is in our favor, that we are getting to meet like great personalities with such great experience and expertise and talk to them. We can discuss our research idea as well with them, and in the break sessions, obviously we can listen to what's actually going around and happening around in the world, what's significant, and how can we proceed and make a career in that particular research area. And then we would be having a talk by Dr. Shindu Hicks. And so I'm just getting a pop-up as participants can now see my screen. I just want to confirm if you all were able to see my screen before as well.
Yes, yes, it was visible.
Okay, thank you. And then we have a talk by Dr. Chris Taylor on bioinformatics, biostatistics, and computational biology. So in my master's, actually, I was told that biostatistics is going to become a very emerging field with great career opportunities. So now what happens with us when we are in our final year, we definitely start thinking about what are the future opportunities, and the field in which we are working. Will we get a stable job or not? Because after COVID, people are facing recession, right? And bioinformatics, biostatistics, and computational biology, these three fields remain those fields which are evolving so much day by day, and these three fields are not going to face any sort of issues. And these fields, obviously, you can see that you can just sit at your home at your own comfort and be a part of such great research around. And the server, especially, you need not have specialized laptops or PCs or systems, and still you can work on your data and get your results and publish a paper just sitting at your home. So that is the beauty of bioinformatics and computational biology, actually. And then we have a talk by Dr. Shamir Kadir. He's from AstraZeneca, and he would be like giving a talk, definitely, and then he would be guiding us as well through what all like a company looks at while hiring people. So job opportunities are the like one of the best things that we are going to discuss about in the symposium, and also the career related opportunities, what a bachelor student should go ahead with or a master student should go ahead with, and what exact skill set a company looks at while hiring candidates. That's a very big question. So in that particular direction, we can like have an expertise and create our skill set, etc., etc. And then we'll have a panel discussion by our panelists, and then the poster presentation, which is on 21st, that would be followed by the panel discussion, and the winners would be announced on 22nd, and that's it. So now please let me know in the chat box what all doubts you have. And also I would love to know from what all fields you are and what is your research interest. You can simply unmute yourself and we can have a word. Okay, so hoping that we have no doubts here. Now, after the workshop, I would be sending you out the guidelines to register yourself for the symposium, because I feel that it's very important that you all can definitely make your weekends work well for you by being a part of such wonderful talks. And the symposium has got three categories. So the first category is for the free listeners. The free listeners can simply join the symposium. They need not register anywhere. And those who are interested to present their research work in the as a poster presentation in the symposium, they need to register as $10. Now, why we have this particular registration fee as $10 is just because we have various prizes that would be given out to the winners of the poster presentation, and also those who want to be a part of their workshops that would be there ahead of the symposium, and also get the basic membership of the OmicsLogic Learn portal. They have to go ahead with the registration fee of $15. I would be sharing the registration page as well with you all, and also the schedule of the symposium and the registration page of the symposium. Now, if you have any doubts, you can mail me at support@pine.bio, or you can also reach out to our marketing team at marketing@omixlogic.com or marketing@pine.bio. Thank you, Sohas, for letting us know about your research interest. And I'll also be giving out a feedback form, and it would be great if you could fill it out for us and let us know about what you feel about the workshop and was your motive accomplished or not. Thank you everyone for joining. We'll be looking forward to meeting you all in the symposium on 21st and 22nd of August. Take care till then. Bye-bye.